Today I will be talking about the case for insect brain emulation. And if you're interested in reading more about this, I recently had a paper published on this in the journal Biological Cybernetics, and I encourage you to check it out. Alright, so why would you want to emulate or simulate the minds of insects? So insects are surprisingly complex. They have complex behaviors and cognition. In particular, bees can count. They can have spatial navigation that's exceptional. They can communicate with their waggle dance language. And then flies are also fairly intelligent organisms as well. And then in addition to this, using insects as an intermediary for the brain emulation of mammals and then maybe eventually humans could lead to sort of a good stepping stone, which would galvanize further research by demonstrating in a very dramatic way that insect behaviors can be recapitulated in silico with using insect brain emulation. And then finally, there are many applications to artificial intelligence, robotics, and then clinical and cognitive neuroscience. So comparing between different states of insect brains may allow us to understand health and disease better, as well as develop biomimetic artificial intelligence and robotics. So first, I'd like to talk just a little bit about mapping the structural neuroanatomy, and it seems that it took away the transparency of some of these images, so that sucks, but whatever. Some of them were translucent so you could see inside, but anyway. So there are a lot of different ways to map structural neuroanatomy, and I won't go into all of these in detail, but suffice to say that there's electron microscopy, which is the highest resolution. There's light sheet microscopy, which requires tissue clearing. Expansion microscopy physically enlarges the tissue to allow for higher effective resolution, as well as the identification of molecular markers within the cells. And then there's X-ray based approaches like X-ray microtomography and X-ray nanoholography. And for insects, while electron microscopy doesn't necessarily scale well to mammals and humans, for insects, electron microscopy is at this point in time fast enough that we've actually already been able to image an entire Drosophila, that's a fruit fly brain, with electron microscopy, and you can see that depicted here. And the tracing of the neurons is currently underway, and they've got sort of a preliminary effort at that going, and it's not yet complete, but folks are well in the process of moving towards that, tracing all the neurons and reconstructing a connectome for the fruit fly. So to emulate the brain, you need to, of course, emulate the neurons. And this is the type of model that would be used for that. It's a multi-compartmental Hodgkin-Huxley type model, and it's actually highly predictive of realistic biological electrophysiology. It's biophysically based on the conductances of different ions across the membrane. These are the equations that are used in it, at least the sort of the core ones. And it operates by breaking up the dendritic tree into these segments that you can see up here. And these segments sort of serve as different compartments that the diffusion of voltage difference across the dendritic tree can be tracked. And so in this way, it's anatomically based, and it very realistically can simulate the electrical activity of neurons. There are also a variety of other details that may be important to include. You wouldn't necessarily need to include every detail, I think, but there are several that might be valuable, and I've listed a few of them here. Due to limited time, again, I'm not going to go into detail on all of these. And then also, we will need to simulate the non-neuronal physiology, so everything besides the nervous system and the brain of the insects. And to do this, there might be possibilities of using a lot of reaction diffusion models to show sort of the diffusion of different neuromodulators and endocrine hormones through the body of the insect. And of course, this is very important for linking the brain and the behavior. And to link the brain and behavior, this will really allow us to see sort of the transition between what is the what is the effect of changes in the brain on behavior, and it'll allow us to sort of dramatically recapitulate how this, this brain behavior link demonstrates the ability to really create virtual insects that can actually behave in silico. So then also I'd like to talk a little bit about hardware and computational resources. So what you see up here is just some calculations that are, there's some details on some calculations that basically the result is that current supercomputers actually should be capable of emulating an entire insect brain, and possibly even the non-neuronal physiology as well, with just the existing supercomputers. And then, because it actually is within the petaflop range, I believe. And that's for both, I did this calculation for both Drosophila, fruit flies, and for honeybees. And another thing that's important to recognize is that there may be emerging neuromorphic hardware that could help increase the ability to do this. Neuromorphic hardware is a circuitry that is structured in such a way as to more closely mimic the nervous system than traditional electronics. And this type of hardware could allow us to, more easily simulate insect brains and other brains as well. All right, so lastly I'd like to discuss the philosophical implications, because obviously as this sort of intermediary with a lot of other applications, but as something that could potentially lead later on to the simulation of human brains, we have to consider the possibility that such brains might be conscious at some point in time. And if such brains were to be conscious, we would want to be very careful with doing experiments on them, because that would essentially entail doing experiments on what would be the equivalent of a living human being. And there could be a lot of terrible things that could be inflicted upon a brain emulation, and we wouldn't want to cause suffering in virtual human beings who have a consciousness or a soul. And so I think that we should definitely investigate, perhaps even using insect brain emulation as a subject of study. We should investigate how consciousness works more deeply before we construct human brain emulations in the farther off future. All right, so with that, I'd like to conclude my presentation and open the floor for questions. Thank you.